Okay, hi everyone. So welcome to another installation of our monthly research reels, although recently it's been twice a month, which is a little treat. But let's get started into a little bit of a preface before we introduce our speaker. So see if this will work for me. So who is IMPART? So IMPART is a national research team headquartered at Dalhousie Medicine, New Brunswick, which encompasses 45 academic and clinical partners across 8 universities. So although we are headquartered in Dalhousie, we are a national collaborating team, and we have many other community partners and funders as well. And Research Reels is a monthly seminar series hosted to improve networking amongst IMPART members, enhance opportunities for future collaborations, and support knowledge translation of our research findings. So it really is aimed to help connect all our laboratories across Canada. And just one more thing, if you want to nominate a speaker, please stay tuned to our post-webinar survey, where you'll also be able to provide some feedback that will really help guide and inform our future webinars. So IMPART is also present on all these different social media platforms where we share accomplishments of our members, our recent publications, these seminar links, and much more. We also have a website, so that's impart.team, where you can visit to see all our members and what their research encompasses. So let's introduce our speaker for today, which is Dr. Alex Quinn, who received his PhD in biomedical engineering from Columbia University, followed by postdoctoral training in Oxford University and Imperial College London. He was recruited to Dalhousie University in 2013, where he is now an associate professor in the Department of Physiology and Biophysics, also cross-appointed to the School of Biomedical Engineering. His research focuses on regulation of the heart's electrical activity by processes that reside within the organ itself, and the role that these autoregulatory mechanisms play in deadly arrhythmias that occur in disease and with aging. He has published 69 papers, 8 book chapters, and 137 conference abstracts, and has been the recipient of numerous awards, including the Faculty of Medicine Award for Excellence in Basic Research from the Dalhousie Medical Research Foundation, the JAF Stevenson Award from the Canadian Physiological Society, and a National New Investigator Award from the Heart and Stroke Foundation. The accomplishment he is most proud of, however, is training the next generation of scientists. In his lab, he is currently supervising numerous postdoctoral, PhD, master's students, and BSc students. So now I'll pass over the floor to the speaker himself.
Thank you very much.
I'll let you share your screen now.
I cannot share my screen. It says cannot start. Okay, because you were sharing.
Yes.
There we go. All right, perfect. So I'm going to go to my slideshow here. I got share sound good. Can you see the pointer? No pointer?
Yes, pointer.
You can pointer. All right, good. All right, thank you for giving me a chance to talk about the research we do in our lab. I am going to make sure this is working. Hold on. There we go. Okay. So we, I chose today to talk a little bit about one of the main focuses of our lab. We end up doing quite a few things around here. So I'm going to focus on one kind of story and then mention a few other things at the end with the spirit of trying to share the broader breadth of what we do in our lab. But I'm going to talk today about, the title that I gave was Tugging at the Heartstrings, the importance of mechanics for electrics in the heart. So I'm hopefully going to impress upon everyone how important it's not just a one-way system in the heart, but it's two ways. So this image here, which is not playing, unfortunately, if you could just give me a moment, I'm going to just try to restart that. Sorry. Now it's playing. I'll share that. Okay, now it's sharing. You can see the, there we go. Do you see the heart beating, everybody? Someone? Okay, hopefully you do.
Yes.
Yeah, all right, great. Thanks. All right, so I like to start every presentation actually with this image, and depending on who I'm presenting to, there's different reasons, but being mostly a cardiac audience and definitely a scientific audience, I show this to you to remind you how dynamic and busy the heart is. So this is a magnetic resonance image of the heart sliced lengthwise down the heart. You can see the ventricles here, the atria, and what you can appreciate is the heart is beating really hard and it's working and doing a lot of work. And it does this constantly throughout your lifetime. 3 to 4 billion times in your life, your heart's going to beat like this. And it doesn't get to rest. Your other muscles, when you go to sleep, rest. Your brain even gets to relatively rest. But the heart keeps going. And this is, not too far off the idea of going to the gym and doing reps for your entire life. So it's a bit of an extreme analogy, but it's not too far off. The heart is generating incredible amounts of pressure. And this is incredible that it can do this for your entire lifetime. And one of the reasons that it works and can pull this off is because it's a very well coordinated and orchestrated organ, where it's basically on a basic level an electrically controlled mechanical pump. So the electrical signals travel in an ordered pattern through the tissue, and that causes the mechanical pumping action. Right? That's what we all understand about the heart. It's fairly straightforward. And this happens, as we know from our physiology classes some of us took, this occurs through excitation contraction coupling. So where intracellular release of calcium inside of the cardiac cells causes interactions of actin and myosin and cross-bridge cycling, which causes the beating to occur. This is your classic textbook understanding of cardiac activity, the most basic fundamental thing that it does. However, the thing that's a little less appreciated, and maybe starting to be a little more, but is that there is a lot of feedback that goes from this mechanical activity back to the electrical activity. And this is what we call mechanoelectric coupling, or sometimes mechanoelectric feedback. And this occurs through stretch activated channels, other mechanosensitive channels, even some mechanosensitive second messenger systems, and definitely mechanosensitivity of calcium handling. So combined, these different mechanisms feedback so that they alter membrane potential and intracellular calcium concentration to affect the electrical activity. Right, and any well-controlled system needs feed forward and feedback to stay in a nice controlled environment. And so this altogether, the other thing we've become interested in is potentially the nerves' involvement in this. But overall, this together creates a mechanoelectric regulatory loop where we have feed forward and feedback, which helps the organ maintain its function. Now, of course, this can be disturbed by external mechanical disturbances, which I'll talk about in a minute. But the important thing to recognize is that all of this is happening within the heart itself. Now, we know that the heart, of course, is modulated and affected by the central nervous system. So if you get startled, your heart will beat faster. That's an autonomic driven response. However, we can transplant the heart from one person to another. We've been able to do that for quite some time. And generally, the nerves don't grow back. On occasion, they do, but typically you do not have reconnection of nerves. Yet someone can live with that heart for 25 to 35 years. And I was just told actually a few days ago that a heart transplant patient has climbed Mount Everest. I wasn't aware of that. But to do that, to climb Mount Everest with a heart that doesn't have normal autonomic control suggests that there is all sorts of auto-regulatory mechanisms going on within the heart itself. And that's kind of the primary maybe interest of myself and the people in my lab, how those autoregulatory mechanisms keep the heart in normal function, and then when they get disturbed, how they may lead to dysfunction. So this is a 1960s scientist depiction of a happy heart, showing us this nice coordinated mechanical activity being driven by the electrical activity. And of course, what you're maybe more familiar with is an electrocardiogram. So we're all familiar with electrocardiogram because in any medical show, you're going to see that in the background on the screen, beeping away, telling us the heart's doing okay. Well, when things go bad, this is a 1960s scientist depiction of a not so happy heart. And you can see this is uncoordinated activity. And this part is not going to be pumping blood well to the body. And of course, on the electrical level, the ECG would look something like this. We'd have that terrible flat beeping sound, just long beep, where there's going to be a lot of people rushing into the room with a lot of equipment to try to save that patient. And I think this audience knows this, but this electrical disturbance is what we call an arrhythmia, okay? There's many causes of arrhythmias. They're associated with many different cardiovascular diseases. We're interested in, though, how these may be driven by these autoregulatory mechanisms. So the idea of having a mechanically induced electrical event or excitation is well known by clinicians. Anyone who puts a catheter into the heart knows that if they make contact with the myocardium with this intracardiac catheter, they can get catheter-induced excitations, right? So that's just the catheter poking the tissue can cause an excitation, well known. And it's actually been known for quite some time in a more extreme case, back since the 19th century, so these case reports from the 1800s, where they saw that there was mechanically induced sudden death, so with a blow to the chest, yet in autopsy, there was no visible evidence of any sort of damage. So it wasn't a traumatic injury, yet these impacts to the chest caused death. And this is what we call commotio cordis, which is commotion of the precordium. So now it's known, it's been seen in sports more commonly now. And so that when you get impacts to the chest, such as with baseballs, hockey pucks, lacrosse balls, or in karate with a knee or an elbow, over the precordium, this impact can result in instant fibrillation. Okay, so ventricular fibrillation, which if not dealt with very quickly, can lead to death. There was a very actually dramatic case of this not too long, and I like to always show this. I mean, I have a Canadian audience here generally today, so but when I'm outside of Canada, I especially like to show this. This was back on Mother's Day in 1998.
Back to the point, Larry Onoff.
I'm going to let this play here. This is a hockey game, and then we'll talk about it.
Back to the point, Larry Onoff up with it there. Larry Onoff hands it across, big drive, and that one blocked and into the.
So this is Chris Pronger for people who are my age. Chris Pronger.
Going to have a two-man advantage.
And Chris Pronger just got hit by a puck. And he has now gone into
ventricular fibrillation. He took the slap shot into the chest.
I'll wait here and play a little lap.
There's a problem here. He took the slap shot into the chest. The trainer's out there quickly, and you can see with that slap shot into his ribs and chest, he's having some problems. And here comes John Wharton from the Detroit bench, just very serious. And then he moved out, and he took a slap shot really high off the chest. Here, right here. Here it comes. Boom, yeah. The left side of his chest near his heart.
So near his heart, that was the thing I was waiting for. So right there, Chris Pronger, just to let you know, 3 days later, he was actually back on the ice and played 3 overtimes. He was a warrior. So, but in that moment, he had gone into ventricular fibrillation. He was in an arrhythmia, and thankfully, they had a lot of medical staff ready and they saved his life. But there's a case of commotion cordis. So that's an extreme case of a mechanically induced event, but there's no actual damage involved. Now, this is the bad side of mechanoelectric coupling. Now, the good side is the potential to use it for therapy. So the defibrillation pioneer, Zoll, back in the 70s, came up with a mechanical pacer where he actually used a staple gun that he modified, and it would allow you to cause impacts to the heart, or to the chest, excuse me, and you could actually pace the heart using this technology. So, I mean, this didn't exactly take off. There is still things called precordial thump, where there's the potential to pace people in asystole, but it's also been used. The ultrasound is another way to mechanically stimulate the heart. So people have shown that more recently that you can use ultrasound to stimulate the heart. So there's still interest in potentially using this as a pro-arrhythmic intervention. Now, but again, going back to clinicians and surgeons, they just intuitively know about the mechanosensitivity of the heart. For here's a heart after surgery, so they are defibrillating the heart. So the heart's been in fibrillation. They're defibrillating the heart, right? There's the paddles in the chest. You'll see in a moment the heart will go back to normal beating. So you can see now the heart is beating away. There it is, a nice rhythm. But in a moment, it's going to go back into fibrillation. And before the surgeon pulls out his paddles again, what he does, you'll see now it's gone back in, he pokes the heart. One poke, give it another poke, come on heart. Third poke, and the heart goes back into normal beating. So intuitively, clinicians are well aware of this mechanosensitivity of the heart, and sometimes even use it to an advantage without maybe even thinking about it. So with this as the backdrop, when I came to Dal, I started the cardiac autoregulation and arrhythmias laboratory. So autoregulation being this internal regulation of the heart, arrhythmias being when it goes bad. And our goal overall is to understand intrinsic regulation of cardiac function and how its dysfunction can lead to deadly cardiac arrhythmias for the development of transformative new therapies. A laudative goal, but generally that's what drives us. Now, I keep saying, us and we. I like to point out now, early in the presentation, that I sit in front of a computer most of the day, as the old guys and gals in the lab tend to do. And the real people who are doing the work are these folks. And so these are the current members of my lab who I'm greatly indebted to for all of the dedication that they have and the hard work that they put in. And so some of the work I'll show is thanks to them, as well as others who've recently left my lab in the last 6 years or so. So the way that we approach research is, we have a particular approach. We have questions that we want to ask, and we try to figure out ways to answer them. And so in my lab, we basically have a toolbox of tools. And when we have a question, we reach into that toolbox and try to figure out a way to address them. So one of our main tools are the zebrafish and the rabbit. So these are most of the work in my lab is done in one of these 2 animal models. Rabbit was my original, what I was trained in, and I like it a lot because its electrophysiology is very similar to human. It's a great arrhythmia model. It's also seen by many as a preclinical model, so it's up the chain enough that it's something that's getting close enough to humans that people think what you find may be relevant. But the zebrafish has become more and more part of my work, and that's because it is a genetic model like mouse. You can do much more high throughput studies typically, but unlike the mouse whose electrophysiology is not so much, at least from ways that we care about, like humans, the zebrafish in a lot of ways is. And it's proven time and time again as I've used it, or those in my lab have used it, to be useful tool to answer fundamental questions that we're trying to ask. And one of the ways we use these is with isolated hearts. So this is an isolated beating zebrafish heart in a dish. This is a rabbit heart, which isn't beating, I'm sorry. But here is us using fluorescence to measure the electrical excitation of the zebrafish heart and the rabbit heart. So we use optical mapping, we call it, or fluorescence imaging to look at voltage and calcium on the surface across the entire surface of the heart. Now, we pair this, sometimes I'm forced to use mouse, or it is the right tool, maybe not even forced. And so sometimes the mouse is useful. We also get tissue from patients now from the surgeons here. So we like to isolate live cells from this tissue and use those as well to see if what we see in the animals is true in the human. Now from the mice, and from our other animal, or from our rabbits and zebrafish as well, we can isolate individual cells. We can mechanically alter those cells, so look at how that affects its activity. And we can look at things, for instance, like calcium sparks, using fluorescence again, to look at how stretch affects the release of calcium inside the cell. And being a biomedical engineer, I sometimes need to at least pretend that I'm an engineer. And so we also like to use models where we can integrate all of this information and ask questions that we can't ask experimentally to drive further research and get new hypotheses. So here, this is just showing a model of fibrillation occurring due to ischemia, much like we're seeing in the rabbit here above. So with all of those tools in the toolbox, the current focuses of the lab here, we have three main projects, funded projects that we're looking at that sort of great umbrellas for what we do. The first is looking at mechanically induced ventricular and atrial arrhythmias, and this has been funded by the Canadian Health Institutes of Health Research, CIHR. We look at autonomic stress-induced arrhythmias in this particular mutation, called POP-DC, and that's been Heart and Stroke funded. And then we look at autoregulation of normal cardiac function. So just how the normal maintenance of cardiac function is maintained, and that's typically been funded by NSERC. So what I'm going to do for the next little bit is talk to you a little bit more about the project we've been looking at mechanically induced arrhythmias, and then briefly mention these at the end as much for a teaser to show you what our other capabilities are. I felt like maybe going through one sort of semi-full story would be a little bit better way to highlight what we do. So we've been very interested in arrhythmias that occur with acute regional ischemia. So when a part of the coronary or portion of a coronary artery is blocked and a region of the heart no longer receives blood flow or the adequate blood flow, that tissue becomes ischemic. So there is a mismatch between the supply and demand of blood and oxygen. And so ischemia is the arrhythmias that occur with ischemia are a major cause of sudden cardiac death. And it's been estimated that 80% of sudden deaths for people who don't have a prior history of heart disease are because of an acute regional ischemic event. Now, this has been investigated, the arrhythmia is investigated for quite some time. This is a classic paper from the late 70s, where they show that in this time period after the beginning of ischemia, so the first 30 minutes in this case, you get these waves of arrhythmias occurring. So this is arrhythmia incidence on the y-axis, and you see you get these waves, and these are just different types of arrhythmias, fibrillation, tachycardia, early excitations that occur through time. So this has been well understood, and there's definitely numerous mechanisms that are involved. But one that has always been implicated but never been well studied is the involvement of stretch. So during ischemia, the tissue here becomes non-contractile, and what starts to happen is the healthy tissue pulls on that tissue, and you start to get stretch of the ischemic tissue, and especially along this border zone. And the magnitude of ischemic stretch has been shown to be a strong predictor of arrhythmias in various studies, and that the probability of arrhythmias are correlated with wall motion abnormalities seen in this ischemic period. So there's been evidence that suggests that mechanics are important, but if it truly is, and the mechanisms that are driving that importance have never really been properly explored, and that's what my first CIHR grant was to look at. So we used our isolated rabbit heart model. Here's an isolated rabbit heart. You can see that you can see here the well, that's actually the vein, but underneath of that, it's a little bit hidden in the tissue, is the one of the main coronary arteries, similar to left anterior descending in a human. Here it's the medial branch of the left circumflex, but it's the equivalent artery in a rabbit. And we put a suture around that artery so that we can ligate it. We have a intraventricular balloon inside the heart, so that allows us to put a mechanical load on the heart because mechanics matters. And we're Langendorf perfusing it, so those who are not familiar, we're retrograde perfusing solution back into the coronary tree, and you can keep a heart beating like this for many hours inside the lab. And so when we tie up that coronary artery, this is just using fluorescent beads to show that we have this region that no longer gets perfused. So this region is the region that will now become ischemic because of our tying off of this ligature here. And so if we use our optical mapping to look at the electrical excitation and repolarization of the heart, here's pre-ligation and 30 minutes post-ischemia, you'll see this region here was much earlier to repolarize. And that's just a classic that's well known for ischemia. Because of a reduction in oxygen, KATP channel, so a ATP-gated potassium channel, turns on, and that causes faster repolarization of the cell. And so when we look at the maps of action potential duration, you'll see there's this region here in blue that's very short, and that's due to the ischemic region. So if we look across from the ischemic to the border, or across the border zone into the healthy tissue, and we look at the action potentials, you'll see that there's this shortening from healthy tissue through to the ischemic tissue. So that's just a classic, I mean, that's well known for ischemia. So this was good for us. We have established ischemic model in the isolated rabbit heart. Well, we wanted to know what was going on with mechanics, because our hypothesis is that it's stretch at the border zone that is leading, or at least contributing, to the arrhythmias. So what we used is we used echocardiography in this isolated rabbit heart, and we looked at different regions around the heart and looking particularly at the border zone of where that ischemia was occurring. So this tissue is all ischemic. This is healthy tissue. This is the border zone. And what we found is that pre-ligation, this is radial strain. So thickening of the heart is, so an increase is actually meaning that you're getting contraction rather than stretch. So here you see proper or normal increasing in thickness of the ventricle as it contracts at the border zone. And then after ischemia, you start to see thinning. All right, so this is instead of it contracting and getting thicker, it's now getting stretched and thinner. So this shows us evidence that we are getting indeed tissue stretch at the border zone. And if we look at our electrocardiogram and our pressure, intraventricular pressure of their balloon, what we see is that we start to see premature ventricular excitations after ligation, as well as more runs of more sustained arrhythmias, such as a ventricular tachyarrhythmia, initially initiated by a PVE that then leads to a sustained arrhythmia. So we have a ischemic model in the isolated rabbit heart where we get stretch of tissue and we see arrhythmias occurring. So we looked at the incidence over time. So this is like that original plot from Koplinski in the 70s that I showed you. If we look at the incidence of arrhythmias over the time after ischemia, here's 0 to 60 minutes on the x-axis, arrhythmia incidence on the y-axis, we see that we get these waves of arrhythmias, very similar to what Koplinski and others have shown, that you get this sort of initial bout and then a later bout. And this involves tachycardias, couplets, and just premature ventricular excitations, which, by the way, have been divided by 10 here. So there's much more of those. And so this is averaged across 10 hearts. And then what we did is we said, well, if mechanics are involved, then if we take away the load from the heart, so we empty that balloon that's inside the heart, so the heart is no longer contracting against the load, that tissue stretch will go away, and we may potentially, if stretch matters, see a reduction in the arrhythmias. And lo and behold, when we unloaded the balloon, what we saw was the arrhythmias almost disappeared. So by taking away that loading, they almost were eliminated. So this really implicated this importance of stretch for the arrhythmias that were occurring. Now we wanted to know a little bit, and we looked at various things. What I'm just going to highlight here is we buffered calcium, intracellular calcium with BAPTA, because we thought calcium might be involved. And that also, in that this is in a loaded heart, so this is with a loaded balloon, but the addition of BAPTA also made the arrhythmias go away. So we felt we have now a case where there is indeed a contribution of mechanics and that it's somehow calcium driven. Well, we wanted then to look at the calcium in the heart. So we adapted our optical mapping setup so that we could excite with 2 different lights, collect 2 different wavelengths, and look at both voltage and calcium. And what we saw here, now you see voltage on the left and calcium on the right. So this is during ischemia, where you're going to see this region that repolarizes early, and then calcium is also, or handling is also affected in that region. And if we look at our action potentials and our calcium transients, so here I show blue is the action potential, and so membrane potential. Red is our calcium transient. Okay, this is through time. And what you'll see is that calcium, is slightly lags behind voltage, and that's what we expect, that, the excitation that causes the release of calcium. But they, fairly close together, they repolarize and come back to resting levels of calcium around the same time. Now, what we found is that in ischemia, because of this shortening of the action potential, and the calcium transient does not shorten as much, we start to get a gap between these. And this leaves a period which is potentially a vulnerable period for arrhythmias, especially calcium-driven arrhythmias, because we have high calcium during a period where the cell is now repolarized and ready to be excited again. So we had a hint, or we thought maybe, our hypothesis was that this elevated calcium in a repolarized cell may be driving the arrhythmias and causing them to occur. So we kind of reached our limit at what we felt we could explore in the whole heart in terms of mechanistic studies. So we then took this to isolated cells. So we have a setup where we can put isolated ventricular myocytes under carbon fibers. So these are very, very thin, smaller than a hair fibers that that adhere to the cell and allow us to either allow the cell to be contracting against a load or to apply a load such as stretch to the heart, to the cell. And at the same time, we can measure voltage and calcium in that cell to see what's happening with its activity. So this is actually, so we can apply stretch. I said we can control the load. So here's a cell under carbon fibers, which we apply stretch, and then we can watch what happens and we can see arrhythmias occur. So what we found is that, so what we did is we established ischemia in these cells. So here's a control case where we have, again, voltage and calcium transient, voltage in blue and the calcium transient in red. We made the cell ischemic. We see much greater shortening of the action potential than the calcium transient. And this leads to this vulnerable period. So this is the difference between the voltage and calcium duration, calcium transient and action potential duration. And so you can see this is an increase in this vulnerable period, potentially vulnerable period, where stretch may cause arrhythmias. And then we can apply stretch to these cells. So this is just looking at sarcomere length. So we're tracking contraction of the cell. And we can see that we can apply stretch either in the diastolic period or in this vulnerable period. So in this period right after the action potential, where that may be potentially highly arrhythmic. And what we see is that we can see premature ventricular contractions, so just single ectopic beats, or more sustained arrhythmias that either resolve or do not resolve depending on the case. So what did we see overall in terms of incidence when we started stretching cells while they were ischemic in these 2 different periods? Well, what we found was that here's just a fraction of stretches that resulted in either a premature ventricular contraction or a more complex sustained arrhythmia. And this is just in normal thyroid, so under control conditions, we have, very few stretch-induced arrhythmias, whether we stretched in diastole or in this vulnerable period. What we found is that in ischemic conditions, there was an increase in stretch-induced arrhythmias, particularly in the vulnerable period. So the vulnerable period had a great increase, and these were actually quite complex arrhythmias, so often sustained arrhythmias that would be more than just a single extra contraction. So ischemia, especially in the vulnerable period, it makes cells more prone to stretch-induced arrhythmias. We then actually did kind of the opposite. Well, what we did is we said, well, if it's this vulnerable period that's so important, let's get rid of that by blocking that KATP channel. So I told you that's what turns on that causes shortening of the action potential. So by blocking that, we were able to reduce this vulnerable period, bring the action potential back to a longer length. And then we redid these stretches and we found that these arrhythmias in the vulnerable period were reduced. So it appears that this vulnerable period really is important in ischemia for causing the arrhythmias with stretch during that period. So this is using a drug called glibenclamide, which blocks the KATP channel. We then looked at the things that we'd seen in the whole heart, for instance, calcium, and we found that buffering calcium with BAPTA, like in the whole heart, also reduced stretch-induced arrhythmias. We got interested in, no time to talk about it right now, why, but a TRP channel called TRPA1, which has been shown to be mechanosensitive, which we accidentally stumbled upon that it may be involved. Blocking TRPA1 greatly reduced the the stretch-induced arrhythmias in the vulnerable period in ischemia, as well as blocking ROS, so reactive oxygen species. So either by blocking the production of ROS or by chelating ROS, mopping it up, that also reduced the stretch-induced arrhythmias. Well, we went one little step further and we said, well, let's go even more subcellular, because we know that stretch increases calcium sparks in cells. That's been shown by others. So here's a cell in control conditions, a cell in ischemia. You'll appreciate there's a lot more calcium sparks. There's a lot more, these are single releases of calcium from ryanine receptors. These sparks, these little flashes of green you can see. In ischemia, there's a lot more to begin with, but with stretch, those are increased in control conditions. And what we found was that in ischemic conditions, so this is the increase in calcium spark rate that's been shown by others that occurs with stretch of cells. The increase in calcium spark rate was greatly enhanced in ischemic conditions. So the normal extra releases of calcium were even greater. And this has been linked to an increase in ROS with stretch. So again, here's the increase in ROS that's been shown before in cells under stretch. And that increase in ROS in ischemic conditions, increase in ROS with stretch was also enhanced. So what we ended up thinking, or what we think we've uncovered, is that this stretch is causing stretch-induced arrhythmias by a TRPA1 calcium and ROS-related mechanism. So just to summarize what we've found in this project so far, the incidence of mechanically induced arrhythmias is greatest in the vulnerable period in ischemia, during acute ischemia. These arrhythmias are dependent on TRPA1, calcium, and reactive oxygen species. And here's our working model, which I won't go through more because I don't have time. But our hope is that these mechanosensitive factors may represent novel antiarrhythmic targets, because they may be contributing enough to the arrhythmias that by targeting them, you can reduce the arrhythmias and make them go away. So that's sort of where we've gotten to very briefly with that project. Where we're going with this now is we're now looking at a more chronic state where we have elevated blood pressure or hypertension. And that in that case, there may be an involvement of TRPA1 as well as microtubules, the skeletal elements inside your cell. And we're looking at the role of these in mechanically induced arrhythmias. And just very briefly, what we found so far is that in ventricular cells, if you activate TRPA1, you get more arrhythmias. If you block it, you get less, or that makes that go away. If you activate TRPA1 but block chelate calcium, it also goes away. And that increasing the amount of microtubules increases the arrhythmias, which are somehow doing that through TRPA1, because again, blocking TRPA1 reverses that effect. And so we've been looking at the density of microtubules with increase of paclitaxel and how these increase arrhythmias in our cells. And we've now moved on to even doing this in atrial cells. So now we're getting either from rabbit or from humans and manipulating microtubule network and other factors to see what's causing stretch-induced arrhythmias in the atria, as that may be very important for atrial fibrillation and other cases. And now we've taken that back to the whole heart. So we've now moved down to the cells, moving back up from what we found in the cells and doing these same interventions, messing with microtubules or increasing microtubules or their post-translational modifications, and seeing how that affects arrhythmias. So this is sort of ongoing work that I'm not going to talk about more now. So I've probably pushed my time to the limit, but I want to just take 2 more minutes to very briefly give teasers about other things we do in the lab, because in the spirit of this being able to share amongst the IMPART members and the idea that we all probably do things that could help each other in our projects, I want just people to know about our other capabilities in case anyone's interested or there are things that might be useful for your own work. So I mentioned that we also look at autonomic stress-induced arrhythmias. So this is our interest in the nerves. And this is where the zebrafish, the rest of this work really is driven by the zebrafish. And the reason I love the zebrafish is because you can see things. This is the isolated zebrafish heart, and you can see the entire intracardiac nervous system in the whole heart. That doesn't work in a mammalian heart. It's too thick. The nerves are embedded in the tissue. It's not possible to do this. And so we can also, of course, zoom in and do much more detailed characterization of the nervous system. But this allows us to, in individual hearts that we've tested, look at how nervous system may be involved in the things we've seen. And so one of the things that postdoc in my lab, Matt Stoyak, has unveiled is this mutation that I talked about. In fish that are healthy with this mutation, here's the nervous system around the pacemaker or the sinus node, and here's the reaction to nervous system stimulation. So you can stimulate the nerves in the isolated heart, and they react normally, they slow, they show bradycardia, and then a post-stimulus tachycardia. However, in hearts that are affected, their nervous system is depleted, it's disorganized, and when you stimulate the nerves, you get arrhythmias, they occur, as is found in patients. So this is a test bed for us now to try to understand these arrhythmias in a way that just does not work in a mammalian heart. Now we're trying to think about how we can now intervene here. So we're trying now to use optogenetics, which is a technique using light and light activated ion channels in the heart or in nerves that can modulate or alter the electrical behavior of nerves and of cardiac cells. So if you've heard of optogenetics, it was, it's a very popular, especially in neurophysiology, technique where we can express light activated ion channels, in this case, in part of the brain, and we can shine light on that region, and then we can see what's happening with heart rate. So we can see how we can alter nervous system activity to affect cardiac function, or we can target the heart itself. So here's the heart inside of, this is a zebrafish larvae. And here we can either shine light on the sinus node and speed up heart rate. You'll see the heart beating away here. When we target the sinus node with light, we speed up the heart, or we can stop electrical activity altogether. Here's the ventricle. And then when we shine light on the ventricle, we can stop it from beating. So this is using light activated ion channels to control the activity. And the hope is that what we can do is, as Matt has shown, that subthreshold stimulation of the nervous system in these mutant fish actually restores activity. So if you drive the nervous system too hard, you get arrhythmias, but possibly just slightly driving the nervous system will in fact restore normal activity. And we've gone even further to try to use this to modulate cardiac activity. And here's a case where early after depolarizations can be eliminated by modulating the action potential, so targeting the cardiac tissue to restore normal activity. So finally, the last thing I'm going to briefly throw at you, it's a lot, but I just want to mention our work where we're looking at autoregulation of cardiac function. And this is where the zebrafish again has become exciting for us, because this is the sinus node of a zebrafish. It's actually a ring, and this is the sinus node of a rabbit. So it's well known since the times of Bainbridge over 100 years ago that if you stretch the sinus node, your heart beats faster. It's an important mechanism, intrinsic autoregulatory mechanism, that allows your heart to deal with an increase in blood flow back to your heart. If you get an increase in venous return, you need to pump that out, so your heart beats faster to do that, so it doesn't explode. So it's known in most all mammals that if you stretch the sinus node, you get an increase in heart rate. So here's stretch and a change in beating rate, except for the mouse. So the mouse slows down. So there's a problem with our genetic model. Yet the zebrafish increases when you stretch the sinus node, much like rabbit or any other mammal that's ever been tested. So we've been looking at ways that stretch is important for sinus node and normal cardiac rhythm. So here's actually a series of zebrafish hearts looking at their beating rate when they're just put into a dish and they're not under normal physiological load. The heart's always under load. And they beat fine, but they're very irregular. They're very erratic in their beating pattern. If you put them under just a little bit of load, similar to what they would be under normal conditions, that beating rate cleans right up and it's much more regular. So there's a baseline mechanical load that's important for normal function of the sinus node. And now we're starting to look at that in development. So Jonathan Bailey in my lab has been cannulating John, has been cannulating zebrafish embryo hearts. So I don't know how he does it, but he gets these tiny needles in there and he then pressurizes the system, which then creates a amount of flow and is able to inflate those hearts, and showing that as the zebrafish develops, so here's 2 days, 6 days, and 14 days of development, you start to see, initially you see no heart rate change with inflation, but then these mechanosensitive mechanisms come on board as development occurs. So that was a lot, but I want to thank everybody for listening. I want to especially thank the current lab members that I showed at the beginning, and as well as past lab members in my group have contributed to this. We have numerous collaborators around the world and also here at Dalhousie who've contributed to this work that we're grateful for, and of course the funding, and I'm happy to take any questions if I haven't taken too long already. So thank you.
No, that was great. Thank you so much, Dr. Quinn. So yeah, we'll open the floor up to the question period now.